Hi there, I'm Dayla Woolnough from Central Michigan University, and today I'll be presenting research on the physiological condition and spatial distribution of unionids, as well as corbiculids in the Laurentian Great Lakes watershed. These are data from Nathan Ring's master's thesis. So, as we know, freshwater mussels are filter-feeding bivalves. They provide a breadth of ecosystem services, including stabilizing sediment in rivers, providing habitat for other organisms, and bioaccumulating chemicals. There are also in huge population declines, likely due to multiple reasons. Especially here in the Great Lakes region, we often think of invasive bivalves as a reason for population declines. Most of the focus of this invasive bivalves in the Great Lakes region are the dreissenids, the zebra mussels and quagga mussels. However, this story that Nathan considered includes the corbicula. They are a genus of invasive bivalves that are found in 4 different forms. Form A is the most common in our area and shown here on the right. Corbiculids were found in the United States in the 1930s and Michigan in the 1980s. This research occurs on the Kalamazoo River. The main branch of the Kalamazoo River is over 200 kilometers in length, and this entire river has a history of anthropogenic contamination, from PCBs to PFAS to one of the largest inland oil spills in 2010. And not much is known about native and invasive bivalves, especially off the main branch in the tributaries. So Nathan asked 2 main questions. Question number 1 was, what are the corbiculid and unionid densities and distributions throughout the Kalamazoo River watershed? And the second question is, what are the physiological associations between corbiculids and unionids in the Kalamazoo River? So we did standardized surveys, timed, quadrat, and scuba surveys for unionids. And after those surveys, we looked for invasive bivalves. These were the sites randomized throughout the watershed based on stream size and temperature. And the surveys that were performed, the blue dots are time searches, orange triangles, time searches and quadrats, because those were sites that had over 25 live individuals or a listed species. And the red squares were scuba surveys that were done in deep water. So to tell you a little bit about the unionid results, we visited 191 sites, and 122 of them, the sites were surveyed. We did time, quadrat, and scuba surveys, as I mentioned before, and we found over 3,400 live unionids of 22 species. The native unionid distribution looks like this, with the larger circles having higher live unionids per person hour, and the blue dots having 0. The green and orange dots are in between. For more information on this, you can scan this QR code or go to this website that we created using a Shiny app, and you can learn more about. I would encourage you to go to Kiera Krishway's talk here at the Freshwater Mollusk Conservation Society conference. For invasive bivalves, we took 1 liter samples, 3 to 5 replicates of those at each of our sites. We dried, sorted, cleaned, and weighed the samples to determine invasive bivalve densities. The corbiculids we put into density categories based on natural breaks. There was control with no corbiculids, low, medium, and high densities. If we extrapolate out to pounds per meter cubed, those were equivalent to 10 pounds per meter cubed, 27 pounds per meter cubed, and 550 pounds per meter cubed. So we had some pretty high density corbiculid sites. If you look at this throughout the watershed, most of the corbiculids were distributed among the main branch of the river, in the Kalamazoo River, with the focus being on the left city, the Kalamazoo, and on the right, or the east, is Battle Creek. The first time that corbiculids were documented in the Kalamazoo was right in between these 2 cities. Interestingly, these 4 green dots, which showed very low densities, the blue dots showing no observed corbicula. Of those 4 dots, there were only 8 valves, which could even be 4 individuals, whereas to the east of that, over 9,000 valves were counted in our samples. So when you look at the corbicular density on the x-axis and the number of unionids found per person hour on the y-axis, you have this positive relationship, which was a little bit unpredicted. As corbiculid density increased, so did unionid density from the number of live found. But we decided to look at those relationships in a little bit more depth. Nathan looked at those with 99% quantile regressions to visualize the potential upper limits of unionid species richness or unionid density. We looked at that by individual unionid species found per person hour, total unionids found per person hour, and unionid species richness. These are just two examples I'm going to show you next on the unionid species on the y-axis found per person hour by corbiculid density, the square root, on the x-axis. This is a negative relationship. So as Urania dilatata increased, corbiculid density decreased. But actinized ligamentina showed an opposite relationship. This was often seen and driven by only a few sites. When we looked at corbiculid and unionid density per person hour, there was a flat relationship when 99th percentile quantile regression was considered. So do unionids and corbicula coexist? This was one of the first questions. Corbicula and unionids do seem to coexist in the Kalamazoo River. Unionid species richness exhibited a negative relationship with corbiculid densities. 13 of the 22 unionid species had a negative association with increasing corbiculid density, and 5 of the 22 unionid species exhibited a positive relationship with corbiculid density. Decreasing unionid species richness with increasing corbiculid density was seen. So that answered our questions. What are corbiculids and unionid densities and distributions throughout the Kalamazoo River watershed? But the second question was, what are the physiological associations between corbiculids and unionids in the Kalamazoo River? To consider the physiological condition, we decided to use Urania dilatata as a surrogate unionid species. We decided on Urania dilatata because it's a common species throughout the watershed and found throughout North America. The impacts between corbiculids and Urania dilatata we determined by using Urania dilatata glycogen levels, Urania dilatata and corbiculid fatty acid signatures, and water quality parameters, which we don't have time to present here today. We sampled a subset of 25 sites of those that were surveyed in 2018 and 2019, shown here by the numbered sites in the map below. To look at unionid physiological condition, first we considered glycogen. Muscles are known to catabolize glycogen under stress, and we could quantify glycogen levels in muscle tissue with a standard method used by the USGS. We collected 87 Urania dilatata from 17 sites, 1 with a control with no corbiculids, 4 sites with low corbiculids, 4 with medium, and 4 with high corbiculids. These are the results. The blue is a control with no corbiculids, and then low, medium, and high as we move from the left to the right on the x-axis. Ellipta dilatata glycogen levels are shown on the y-axis. As you can see, the control had a higher glycogen level overall, but due to the variation, there were no significant differences in these relationships. So, when we look at Urania dilatata's physiological condition, glycogen levels did vary in Urania dilatata by corbiculid density, also in stream size and water temperature, which is not shown here. But the variation in glycogen levels were not statistically significant. Glycogen is known to be highly variable in other research as well. Then we decided to look at Urania dilatata and corbiculid diet. We can consider fatty acids when looking at short-term and long-term diets. And by comparing fatty acid signatures between Urania dilatata and corbiculids, we can look at the hypothesis that there's a potential competition for food between corbiculids and unionids. For our fatty acid analysis, we collected tissues in the summer of 2020, therefore the reason we're always wearing masks. And the tissue from corbiculids and Urania dilatata were collected at 20 sites, corbiculid only, unionid only, low, medium, and high corbiculid density. These tissues were sent to Dr. Jacques Rincard at SUNY Brockport, and he helped us to look at the potential diet overlap, considering 31 fatty acids. This is an NMDS where each point represents corbiculid or Urania dilatata sample. The blue ones are corbiculids, the green ones are dilatata. when considering all 31 fatty acids, there was a distinct relationship that showed that they were really different fatty acid signatures in these two different groups. The fatty acids separation was due mostly in part to these 6 species of fatty acids shown here in the first column on the left. The 2 that are circled in or have red squares around them are ones that are fatty acid species that are known to represent green algae. When monosaturated, polyunsaturated, and saturated fatty acids were considered in the same way, we also see the split between corbiculids and unionid dilatata, with the unionids representing more of the monounsaturated species of fatty acids, and the corbiculids being more the polyunsaturated and saturated fatty acids. When we looked at size of stream, we saw the same relationship shown by the red near the bottom, green in the middle, and blue near the top between both groups of unionids and corbiculids. So we still see that split, but we see a gradient along NMDS axis 2 between size of river. So unionid dilatata and corbiculid diets have unique fatty acid structure. This shows that corbiculids and unionid dilatata fatty acids have little overlap, so there's a potential lack of competition that was previously hypothesized. Fatty acids are commonly found in algae, and we see that these mussels are likely feeding on the green algae, which is not a surprise, but documented here. So to summarize, corbiculid invasion in the Kalamazoo River. Corbiculid invasion is likely ongoing because we have this origin between Battle Creek and Kalamazoo, but little in the downstream area of the Kalamazoo. Glycogen levels in unionid dilatata showed no relationship with corbiculids, and there was limited overlap in fatty acid structures between unionid dilatata and corbiculids, so perhaps less competition for food than previously thought. For our future directions, we hope to use the shell data from the surveys as well as growth lines to determine changes over time, and to consider the impacts of corbiculids on other unionid species in controlled and field settings. So with that, I'd like to acknowledge all members of the Woolnough and Zanata teams that played a role in these surveys from 2018, 2019, and 2020, as well as Joe Rathbun that helped us with our surveys, Dr. Kevin Pangol and Hunter Carrick that were on Nathan's committee, especially Dr. Jacques Rincard from SUNY Brockport that helped with the fatty acid analysis, funding from the Kalamazoo River Community Recreational Foundation, CMU and the Institute for Great Lakes Research, and Golder Associates, where Nathan is working now. So with that, I'd be happy to answer any questions during the question period, or feel free to reach out to myself or Nathan via our email and scan, if you're interested in the Kalamazoo data, the QR code on the top left, and feel free to follow us on Twitter and Instagram.